So hello again. So as you may have seen by now, radiopharmaceuticals have certain peculiarities that make them different from other drugs. So during this presentation, I want to focus on these differences and the implications they may have when conducting clinical studies. So why are radiopharmaceuticals different from other drugs? Well, there are two main differences that I want to highlight. First of all, radiopharmaceuticals are injected in very low concentrations. And secondly, due to their radioactive nature, they only exist for a limited period of time, which is dependent on the half-life of the radionuclide present in that radiopharmaceutical. Now, let's focus on the first aspect, the low concentration. Radiopharmaceutical development shares similarities with drug discovery, but there are key differences that allow a faster, safer, and more cost-effective route for taking radiotracers into human studies. One main difference is that in PET and SPECT imaging, the dose or the concentration of the administered radiotracer is always below the pharmacologically active dose and must never produce a biological response. So this is called microdosing, where the administered dose is below 100 micrograms with no pharmacological effects. As a result, PET or SPECT radiopharmaceuticals only require very basic toxicological assessment before progressing into first-in-human studies. So this significantly shortens development time and the costs, because toxicology studies can be very expensive. They are generally around 100K. So due to this microdosing, the regulatory authorities have cut down the requirements for preclinical safety testing. However, this does not actually mean that radiotracer development is cheap, because as you've seen before, radiotracers need to be produced to GMP standards and require specialist equipment such as a cyclotron and radiochemistry laboratories. As they are injected in very low concentrations, the side effects of PET and SPECT tracers are very uncommon, and the concern of toxicity is minimized. Besides, certain aspects that are very important for other drugs, such as oral bioavailability and therapeutic window, do not apply to PET and SPECT tracers. Now, let's go back to how tracers are made. So you will remember from the previous talks that the first thing that we need to do is obtain the radioactive source, the radionuclide. The concentration of this radionuclide ranges from nano to picomolar. So as you can see, it's very, very low. We then mix this radionuclide with the molecule we want to radiolabel, which is called the precursor, and it's represented by the blue circles here. And we add any other required reagents and excipients represented by the green and the gray circles in this picture. So all these components are in much higher concentration than the radionuclide, so they are generally in the millimolar range. So what's the consequence of this? Well, as the concentration of the blue precursor circles is higher than that of the radionuclide, at the end of the reaction, only a few blue circles will be radiolabeled with the radionuclide, and the rest will remain not labeled. Now, in some cases, this whole mixture is injected into the patient. So all these components are injected. And this is generally the case for radiotracers made from generator-produced radionuclides and kits, like Jen explained previously in her presentation. But for radiotracers produced in a cyclotron, there is generally a purification step to get rid of any unlabeled molecules and other unwanted reagents as part of the synthesis. So you end up with something more like this. Now, regardless of which option is injected into the patient, either this one or this one, you can see that the concentration of the radiolabeled molecule is always very, very low. It's in the nano to picomolar range. So what consequences or implications does this low concentration have? Well, firstly, as I have already said, these tracers do not produce pharmacological effects. But also, due to their low concentration, radiopharmaceuticals cannot be tested or characterized or analyzed using standard techniques that would be used to characterize any other drugs. For example, you cannot use NMR spectroscopy or mass spectrometry, because these techniques, even though they can determine the structure of the molecule, they require higher concentration of the molecule, which is what we don't have with radiotracers. So, as we've seen in the previous slide, the drug substance, that is the active ingredient or the radio-labeled molecule, is not isolated from the drug product, from the whole formulation that is going to be injected. So this also makes the characterization of the radio-labeled compound more difficult. So this is why the drug substance is never tested on its own. It is the final formulation to be injected, the drug product, which gets tested. So if you are using a radiopharmaceutical as an IMP in a clinical trial, so you will have an IMPD, an IMP Dossier, which is a document that summarizes information related to the quality, manufacture, and control of the radiotracer. Now, IMPDs generally have a defined structure, and this document has separate sections for the drug substance and the drug product. But for radiotracers, the section regarding the characterization of the drug substance, this one, will be pretty empty and mainly will just refer to the section of the characterization of the drug product, because this is what gets tested. So how do we check the identity and purity of the active ingredient within the final formulation? Well, we do this by comparison with the non-radioactive equivalent of the radiotracer. So if our radiotracer is compound X labeled with F18, then the non-radioactive equivalent would be compound X with the non-radioactive fluorine-19 in place of the F18. Similarly, if the tracer is compound X with radio-labeled with carbon-11, then the non-radioactive equivalent would be compound X, but with the non-radioactive carbon-12 in place of the carbon-11. So as opposed to the radioactive compound, the non-radioactive equivalent compounds can be purchased or synthesized in higher quantities, so they can be fully characterized using techniques non-radioactive compounds would normally use, such as NMR or mass spectrometry. Then, after we have done this, using HPLC, we see how the compounds in the drug product behave compared to the non-radioactive reference. So the HPLC will display two measurements, one being the absorbance or conductivity, and the other one being the radioactive measurement. Now, the non-radioactive reference standard will only show results for absorbance or conductivity, but not for radioactivity. In the radioactivity window, we will just see noise, but in the other window, we will see a signal. So here we can see that the reference standard shows a retention time of about 26 minutes. So in theory, our radioactive compound should have the same retention time. So when we test our formulation, the drug product, we can see that the non-radioactive chromatogram shows various peaks here, which will correspond to the other reagents or excipients present in the formulation. Because remember that the concentration of the radioactive compound is so small that will not show here. But in the radioactivity window, however, we see there is something radioactive that has a retention time of about 26 minutes, as expected. So this is good. So finally, what we do is we mix the drug product and the reference standard, and then we test this mixture. So here we can see that both the radioactive compound and the non-radioactive reference standard show the same retention time. So here, with this, we can be pretty confident that the structure of our radioactive molecule is correct. So as we've said before, due to their short half-life, radiopharmaceuticals must be manufactured on the day of administration. Manufacture is not always simple, and failure to meet the required specifications is not rare. Apart from potential hazards of radiopharmaceutical production, the short half-life forces QC procedures to be performed very quickly. Therefore, Annex 3 of the GMP guidelines allow the release of radiopharmaceuticals before completing all QC tests, and it allows radiopharmaceuticals to be administered to patients prior to performing sterility testing. So radiotracer production occurs in a non-sterile environment in which microbial contamination is controlled and minimized through adherence to strict operating procedures. And the sterility of the pharmaceutical is ensured by filtration of the drug product through a 0.2 micron filter. However, the integrity of this filter, which is assessed after the filtration has happened, is not always tested before the radiopharmaceutical is injected to the patient either. Nowadays, in most cases, the filter integrity test is done in an automated way, and therefore the results are available before releasing the tracer. However, in laboratories where this is tested manually, the results may not be available before release due to the need to protect the operators from receiving a high radiation finger dose by performing the test. Before the tracer is released, as Jen mentioned in her presentation, radiopharmaceuticals are tested for endotoxins, which can cause a systemic inflammation response. However, the injection of a product, the sterility of which is not confirmed, is always far from ideal. So in these cases, as specified in Annex 17 of the GMP guidelines, the manufacturing process of the radiotracer must be validated to prove that the required specifications of the final product, including sterility, can be achieved consistently. As the filter used for the filtration of the tracer is the only mechanism in place to make the final product sterile, it is very important that process control and risk mitigation measures are implemented during the manufacturing process. Now, some of these measures include testing the bioburden throughout the manufacturing process and minimizing the microbial load to the sterility filter that is used during the production. But despite these risk mitigation measures, sterility failure cannot be ruled out prior to tracer administration. And this obviously can have serious consequences because most of these patients are also immunocompromised. On the other hand, any failure to meet any of the pre-release specifications would result in the tracer not being released for patient use. And although this failure cannot be anticipated, at least it is easily picked up before any harm is caused to a patient. As radiotracers are prepared right before their administration, patients will have already arrived to the nuclear medicine department before the end of the synthesis, and therefore before any QC checks are carried out. So consequently, QC failures would cause the inconvenience to the patient who have traveled to the hospital and have the scan cancelled or rescheduled as a consequence of the QC failure. So when such failures happen, the research team must be open and transparent with patients and explain that these things unfortunately do happen. Obviously, any QC failure should trigger an investigation to identify the root cause of the failure and the implementation of corrective and preventative actions where applicable. And in cases where there has been a pre-release QC failure, it may not always be possible to repeat the synthesis. So let's use the example of a PET radiotracer, which, as we have seen before, is synthesized using a module that sits inside a hot cell. So at the end of the synthesis, this synthesis module will be very radioactive. So the operator cannot just open the hot cell, clean the synthesis module, replace the reagents, and prepare it for the next synthesis. Because usually you need to wait about 10 half-lives before opening that hot cell, depending on the amount of radioactivity you used in the synthesis. So if this is an F18 tracer, which has a 2-hour half-life, this means that you cannot open the hot cell until the next day. So if there is a tracer failure, then you cannot repeat the synthesis on the same day. Now, let's use a practical example of the manufacture of a radiopharmaceutical IMP used in a clinical trial. So in this case, the tracer is F18 labeled tetrafluoroborate, PF4. So after the synthesis, pre-release QC checks will be carried out and a certificate of analysis will be issued, like this one you see here. This certificate of analysis will list the QC tests required for this tracer, the release specifications, and the test results. So as you can see here, the table shows the results of all the tests except those of the sterility testing and the filter integrity testing, which in this case was being done manually the day after release to protect the operators from receiving a high radiation dose. So the results will be checked by QC personnel, and in this case, as the radiotracer is an IMP, also by a QP. The QP will also sign a release certificate, but given that not all QC test results are available at this stage, this release certificate signed at this stage is only a conditional certification of the radiotracer. So this document will contain information such as the radioactivity in the released vial, the time of release, the expiry time, and the maximum volume that can be injected to the patient. The document will also state that the batch is being released following satisfactory completion of the first stage QC testing and still pending results of the filter integrity testing and sterility. This is why this document is part 1 of 2. So if all the pre-release tests are satisfactory, the tracer will be injected to the patient. Now, the following day, the radioactivity in the filter used to make that radiotracer sterile will have decayed to safe levels for operators to test it. So they will do the test. So what happens if these filter integrity test results fail to meet the specifications? Well, if you suspect you've done the test wrong, you may be able to repeat the test. But if the test fails again, then you can be pretty confident that the integrity of the filter has been compromised. Now, this increases the risk of the radiotracer being non-sterile when it was injected to the patient. So you now need to wait to receive the sterility results, which take approximately 4 weeks. So in the meantime, a deviation should be raised for the filter integrity test failure. And if the sterility results are fine, then filter integrity failure, even though it will need to be investigated, is not likely to have caused any issues to the patient. But what if the sterility results fail to meet the specifications and show that the tracer was not sterile? Well, if both the filter integrity and the sterility tests fail to meet the specifications, then it is most likely that it is a true failure and the tracer was not sterile at the time of administration. Therefore, a deviation must be raised and an investigation must be initiated to find out the root cause of the failure and implement any appropriate actions. So the results must be reported to the CTU and the sponsor, and then we need to find out if there have been any adverse events or adverse reactions reported. Besides, manufacturing of that tracer for human administration will have to stop until 1 or more synthesis test runs are done, showing that all QC specifications can be met. Now, on the other hand, if the filter integrity test has passed, but the sterility results fail, then we have to first question if this is a true failure or if there were any issues when performing the sterility test. Therefore, the first thing to do is to carry out an investigation about the test. So this will include aspects such as, was there an obvious error during testing, such as testing equipment failure, a power outage, or something similar. But regardless of the testing error being identified or not, there generally isn't any leftover sample to repeat the test. Therefore, we should always proceed as if the result is a true failure and do all those things that I just mentioned earlier, such as report the failure to the CTU and the sponsor, find out if there have been any adverse events or reactions, etc. So when the post-release QC tests are completed, the QP will sign the second part of the release certification. And this will be approximately 1 month after the injection of the radiotracer to the patient. And to finish the presentation, I just want to mention the differences when manufacturing radiotracers as IMPs for clinical trials or as non-IMPs for other clinical studies or routine care. The Medicines for Human Use clinical trials regulations require that all IMPs are manufactured according to GMP at sites that hold an MIA IMP license and that the IMP batches are released by a qualified person. But the majority of the radiotracers used within the NHS are not IMPs used for clinical trials. Instead, they are mainly manufactured within NHS radiochemistry and radiopharmacy facilities under a specials license following Regulation 167 of the Human Medicines Regulations 2012. A specials license is needed to manufacture medicinal products without a marketing authorization, that is, products that are not licensed. So there are certain restrictions that come with products made under a manufacturer's specials license, such as, for example, products must be used for clinical special needs of an individual patient as determined by the prescriber, and you cannot advertise or market these products. So as explained in the MHRA guidance, a qualified person is not required for the release of an unlicensed product under a specials license, and therefore most hospitals do not have the services of a QP permanently at their disposal. So again, this is one of the reasons why not every site within the NHS can manufacture radiotracers as IMPs for clinical trials.